Next story. Unleashing the power of Medusa's petrifying stare. In a groundbreaking study published in Nature Chemical Biology, scientists have developed a new analytical method that can identify genetic determinants of cell death, offering promising implications for cancer research and drug discovery. The study, led by researchers at the Icahn School of Medicine at Mount Sinai, presents a novel approach to chemogenetic profiling, a technique used to reveal genetic determinants that coordinate phenotypic responses to therapeutics. Traditional methods of chemogenetic profiling, however, primarily measure changes in cell proliferation and can conflate the biology that regulates proliferation and cell death outcomes. To address this limitation, the researchers developed MEDUSA, method for evaluating death using a simulation-assisted approach, an analytical method that integrates experimental data with computational models to deconvolute drug-induced changes to both cell proliferation and cell death rates. MEDUSA allows us to infer a death rate from measured clonal abundance and calculated proliferation inhibition data, explains Jesse D. Gelles, co-author of the study. By leveraging these data and experimentally derived models, MEDUSA can specifically identify death regulatory genes that underlie the drug response. The researchers demonstrate that traditional chemogenetic profiles measuring relative viability are more sensitive towards detecting proliferation regulatory genes while failing to identify death regulatory genes. MEDUSA, on the other hand, accurately identifies death regulatory genes using simulations of drug-induced population dynamics constructed from a plate-based assay for measuring and parameterizing live and dead cell populations over time. As a proof of concept, the researchers studied DNA damage-induced death in the presence or absence of TP53, a commonly mutated tumor suppressor gene that induces both cell cycle arrest and pro-apoptotic signaling downstream of DNA damage. The study revealed that loss of TP53 led to increased rates of drug-induced death, in apparent contradiction with prior studies measuring viability or apoptosis. Experimental and genetic approaches, combined with MEDUSA analysis, revealed a unique dependency upon the electron transport chain and mitochondrial oxidative phosphorylation in p53-deficient cells treated with the conventional chemotherapeutic agent, etoposide, says Jerry Edward Chapuk, co-author of the study. The researchers further determined that the DNA damage-induced cell death in p53-deficient cells is dependent on high NAD+ levels, and drug-induced lethality was rescued by genetic or pharmacological reduction of the NAD+ pool. The study highlights the bias in chemogenetic profiles based on clonal abundance and demonstrates MEDUSA's capability to score the death regulatory function of genes. The researchers suggest that MEDUSA can aid in characterizing forms of non-apoptotic cell death that remain poorly understood and contribute to broader human diseases and therapies. MEDUSA is a promising analytical method for chemogenetic profiling efforts, and its ability to determine death rates through extrapolation without requiring recovery of dead cells makes it particularly suitable for models that undergo non-apoptotic cell death, says Gelles. The study's findings lay the groundwork for future investigations into the role of MEDUSA in various drug mechanisms and non-apoptotic cell death, providing valuable insights into the complex interplay between genes, drugs, and cell death regulation. Source: Gelles, J.D., Chapuk, J.E. The Death Gaze of MEDUSA. Nat Chem Biol, 2024. Less than DOI greater than. Next story. Combating Toxic Lab Culture, Lessons from Superconductivity Case. In a recent Nature correspondence, journalist Juan Pablo Fuenzalida Werner highlights the concerning issue of universities neglecting the welfare of PhD students in their investigations of research misconduct. The case of superconductivity researcher Ranger Diaz, whose university failed to consider the impact of his behavior on his PhD students, is not an isolated incident. The lack of feedback mechanisms for assessing the impact of researchers on their PhD students allows them to gain recognition for their work while creating adverse conditions for their students. At best, this gap risks alienating academically gifted students. At worst, it fosters an environment ripe for scientific misconduct. Graduate schools must recognize and act on this issue by conducting regular, anonymous interviews with PhD students to detect and address any form of harmful behavior by senior researchers. A PhD is a challenging experience, but it should never be a training course in survival in a toxic environment. Universities and funding agencies should have zero tolerance for laboratories with an abusive culture. By transferring knowledge and motivating the next generation, laboratories with a positive culture can inspire and encourage academic growth. This issue highlights the need for a cultural shift in academia, where the welfare of PhD students is prioritized, and toxic lab cultures are not tolerated. By taking proactive steps to detect and address harmful behavior, graduate schools can create a positive and supportive learning environment for PhD students. Source: Superconductivity case shows the need for zero tolerance of toxic lab culture. By Juan Pablo Fuenzalida Werner. Nature 627, 735, 2024, DOI. This story is 350 words long. Next story. Unveiling the construction of Sydney Harbour Bridge a century ago. As the world commemorates the 100th anniversary of the Sydney Harbour Bridge, it's fascinating to take a trip down memory lane and revisit how the iconic landmark was taking shape a century ago. According to an article published in Nature on March 29, 1924, the contract for the bridge had been placed, and some intriguing details had emerged about its design and construction. The bridge was to be an engineering marvel, with a single arch of 1650 feet span, making it the largest arch bridge in the world. The total length of the steel arch and approach spans was to be 3,770 feet, dwarfing the nearest competitor, which had a span of about 1,000 feet. The bridge's massive scale was necessitated by the need for rail and roadway accommodation, resulting in a total width of 150 feet. The main span was to be of exceptionally massive construction, and it was believed that the bridge would have the heaviest single span in the world. The manufacturing of the heavy steel plates required for the bridge's construction was to take place in Middlesbrough, with the whole of the material being assembled in special workshops on the site. The bridge was a testament to the engineering prowess of the time, and its completion was eagerly awaited by the people of New South Wales. Coincidentally, an article published in Nature on March 29, 1974, highlighted the scientific progress made in the field of superconductivity, which was discovered in 1911. The article reviewed a book titled Superconductive Tunneling and Applications by L. Solomar, which provided a comprehensive overview of the first decade of research on superconductive tunneling. The book was described as a Michelin green guide to the field, offering a 2-paragraph introduction to the history of a particular chateau and a complete reference list of nearly 103 entries for the reader who wanted to delve deeper. The review concluded that the book was an excellent resource for anyone interested in the field of superconductivity and its applications. The two articles, published 50 and 100 years ago, respectively, provide a fascinating glimpse into the scientific and engineering achievements of their time. The Sydney Harbour Bridge, which was still under construction a century ago, has become an iconic landmark and an enduring symbol of human ingenuity and perseverance. At the same time, the progress made in the field of superconductivity, as detailed in the 1974 article, has led to significant advancements in various fields, including physics, engineering, and technology. Sources: 1. The contract for Sydney Harbour Bridge, New South Wales, has been placed. 1924. Nature. 113, 2846, 416. DOI. 10, 1038 113416 AO. 2. Superconductive Tunneling and Applications, 1974. Nature. 248, 189. DOI. 10, 1038 248189 AO. Next story. Unveiling the truth behind the widespread mother tree concept. In the world of forest ecology, a heated debate has emerged about the idea of the mother tree, the concept that mature trees in a forest, or mother trees, help nurture and support younger trees through an underground network of fungi. The idea, popularized by forest ecologist Suzanne Simard, has captured the public's imagination and inspired a new appreciation for the complexity and interconnectedness of forests. However, some scientists argue that the scientific basis for the mother tree idea has been oversold. At the heart of the controversy is the question of just how extensive and significant the underground network of fungi connecting trees in a forest really is. While it is well established that trees can and do form symbiotic relationships with fungi through their roots, the idea that this network extends to form a vast, interconnected web of support and communication between trees is still a topic of active research and debate. Some scientists argue that the idea of the mother tree has been overhyped in popular accounts, and that the evidence for the extent and significance of the underground network is not as clear-cut as some have suggested. They point out that while there is evidence of carbon and nutrient transfer between trees through fungal networks, the extent and significance of this transfer is still a matter of debate. Furthermore, they argue that the idea of mother trees actively nurturing and supporting the young through this network is not well supported by the evidence. On the other side of the debate, supporters of the mother tree idea argue that the network of fungi connecting trees in a forest is a crucial aspect of forest ecology that has long been overlooked. They point to a growing body of evidence suggesting that the network is extensive, complex, and vitally important to the health and resilience of forests. They argue that the idea of mother trees actively nurturing and supporting the young is a useful and intuitive way of thinking about the network, and that it has helped to generate interest and support for forest conservation efforts. At the center of this debate is Suzanne Simard, the ecologist who first popularized the idea of the mother tree. Simard is a passionate and outspoken advocate for the importance of forests and the need to protect and preserve them. She argues that the mother tree idea has helped to generate interest and support for forest conservation efforts, and that it has challenged the traditional view of forests as competitive, dog-eat-dog environments. However, Simard has also faced criticism from some scientists who argue that the mother tree idea has been oversimplified and overhyped, and that it risks misleading the public about the true nature of forest ecology. They argue that the idea of mother trees actively nurturing and supporting the young through the network is not well supported by the evidence, and that it risks distracting from the more complex and nuanced reality of forest ecology. So where does the truth lie? It's clear that the network of fungi connecting trees in a forest is a complex and fascinating aspect of forest ecology, and that there is still much that scientists have to learn about it. While the idea of mother trees actively nurturing and supporting the young through the network is a compelling one, it's important to approach it with a critical eye and a nuanced understanding of the evidence. Ultimately, the debate over the mother tree idea serves as a reminder of the importance of careful, evidence-based science in shaping our understanding of the natural world. As we continue to explore and learn about the complex and fascinating world of forest ecology, it's crucial that we approach the evidence with an open mind, a critical eye, and a commitment to rigorous scientific inquiry. Source: Less Than Nature Greater Than. Next story. Empowering Climate Predictions with AI Technology. Next story. AI revolutionizes climate modeling.